Chemical reactions. The objective of this chapter is to define a chemical reaction, describe the various forms of energy, compare exergonic and endergonic chemical reactions, describe the role of activation, energy, and catalysts in chemical reactions, describe synthesis, decomposition, exchange, and reversible reactions. A chemical reaction occurs when new bonds form or old bonds break between atoms. Chemical reactions are the foundation of all life processes, and, as we have seen, the interaction of valence electrons are the basis of all chemical reactions. Consider how hydrogen and oxygen molecules react to form water molecules. The starting substance, 2H2 and 1O2, are known as the reactants. The ending substances, 2 molecules of H2O, are the product. The arrow in the figure indicates the direction in which the reaction proceeds. In a chemical reaction, the total mass of the reactants equal the total mass of the products. Thus, the number of atoms of each element is the same before and after the reaction. However, because the atoms are rearranged, the reactant and product have different chemical properties. Though thousands of different chemical reactions, body structures are built and body functions are carried out, Through thousands of different chemical reactions, body structures are built and body functions are carried out. The term metabolism refers to all of the chemical reactions occurring in the body. Forms of energy in chemical reactions. Each chemical reaction involves energy changes. Energy is the capacity to do work. Two principal forms of energy are potential energy, energy stored by matter due to its position, and kinetic energy, the energy associated with matter in motion. For example, the energy stored in water behind a dam or in a person poised to jump down some steps is potential energy. When the gates of the dam are opened or the person jumps, potential energy is converted into kinetic energy. Chemical energy is a form of potential energy that is stored in the bonds of compounds and molecules. The total amount of energy present at the beginning and end of a chemical reaction is the same. Although energy can be neither created nor destroyed, it may be converted from one form to another. This principle is known as the law of conservation of energy. For example, some of the chemical energy in the food we eat is eventually converted into various forms of kinetic energy, such as mechanical energy used to walk and talk. Conversion of energy from one form to another generally releases heat, some of which is used to maintain normal body temperature. Energy transfer in chemical reactions. Chemical bonds represent stored chemical energy, and chemical reactions occur when new bonds are formed or old bonds are broken between atoms. The overall reaction may either release energy or absorb energy. Exergonic reactions, X meaning out, release more energy than they absorb. By contrast, endergonic reactions, end equals within, absorb more energy than they release. A key feature of the body's metabolism is the coupling of exergonic reactions with endergonic reactions. Energy released from an endergonic reaction often is used to drive the endergonic one. So to repeat that, a key feature of the body's metabolism is the coupling of exergonic reactions and endergonic reactions. Energy released from an exergonic reaction often is used to drive an endergonic one. In general, exergonic reactions occur as nutrients such as glucose are broken down. In general, exergonic reactions occur as nutrients such as glucose are broken down. Some of the energy released may be trapped in the covalent bond of adenosine triphosphate, also known as ATP, which we describe more fully later in this chapter. In a molecule of glucose, If a molecule of glucose is completely broken down, the chemical energy in its bonds can be used to produce as many as 38 molecules of ATP. The energy transferred to the ATP molecule is then used to drive endergonic reactions needed to build body structures, such as muscle and bone. The energy in ATP is also used to do the mechanical work, which is involved in the contraction of muscles or the movement of substances into or out of our cells. Activation energy. Because particles of matter, such as atoms, ions, and molecules, have kinetic energy, they are continually moving and colliding with one another. A sufficiently forceful collision can disrupt the movement of valence electrons, causing an existing chemical bond to break or a new one to form. The collision energy needed to break the chemical bonds of the reaction is called the activation energy of the reaction. Thus, initial energy investment is needed to start a reaction. The reactions must absorb enough energy for their chemical bonds to become unstable and the valence electrons to form new combinations. Then, as new bonds form, energy is released to the surroundings. Both the concentration of particles and the temperature influence the chance that a collision will occur and cause a chemical reaction. Concentration. The more particles of matter present in a confined space, the greater the chance that they will collide. Think of people crowding into a subway car at rush hour. The concentration of particles increases when more are added to a given space or when the pressure on the space increases, which forces the particles closer together so that they collide more often. Temperature. As temperature rises, particles of matter move about more rapidly. Thus, the higher the temperature of matter, the more forcefully particles will collide, and the greater the chances that a collision will produce a reaction. Catalysts. As we have seen, chemical reactions occur when chemical bonds break or form after atoms, ions, or molecules collide with one another. Body temperature and the concentrations of molecules in body fluids, however, are far too low for most chemical reactions to occur rapidly, enough to maintain life. Raising the temperature and the number of reacting particles of matter in the body could increase the frequency of collision and thus increase the rate of chemical reactions, but doing so could also damage or kill the body's cells. Substances called catalysts solve this problem. Catalysts are chemical compounds that speed up chemical reactions by lowering the activation energy needed for a reaction to occur. The most important catalysts in the body are enzymes, which we will discuss later in this chapter. A catalyst does not alter the difference in potential energy between the reactants and the products. Rather, it lowers the amount of energy needed to start the reaction. For chemical reactions to occur, some particles of matter, especially large molecules, not only must collide with sufficient force, but they must hit one another at precise spots. A catalyst helps to properly orient the colliding particles. Thus, they interact at the spots that make the reaction happen. Although the action of the catalyst helps to speed up the chemical reaction, the catalyst itself is unchanged at the end of the reaction. A single catalyst molecule can assist one chemical reaction after another. Types of chemical reactions. After a chemical reaction takes place, the atoms of the reactants are rearranged to yield products with new chemical properties. In this section, we will look at the types of chemical reactions common to all living cells. Once you have learned them, you will be able to understand the chemical reactions so important to the operation of the human body that are discussed throughout the book. Synthesis reactions, or anabolism. When two or more atoms, ions, or molecules combine to form new and larger molecules, the processes are called synthesis reactions. The word synthesis means to put together. A synthesis reaction can be expressed as follows. A. Atom, ion, or molecule A plus B. Atom, ion, or molecule B combine to yield new molecule AB. One example of a synthesis reaction is the reaction between two hydrogen molecules and one oxygen molecule to form two molecules of water. Another example of a synthesis reaction is the formation of ammonium from nitrogen and hydrogen. N2, one hydrogen molecule, plus 3H2, three hydrogen molecules, combines to yield 2 NH3 or 2 ammonia molecules. All the synthesis reactions that occur in your body are collectively referred to as anabolism. Overall, anabolic reactions are usually endergonic because they absorb more energy than they release. Overall, anabolic reactions are usually endergonic because they absorb more energy than they release. Combining simple molecules like amino acids, discussed shortly, to form large molecules such as proteins is an example of anabolism. Decomposition reactions or catabolism. Catabolism reactions split up large molecules into smaller atoms, ions, or molecules. A decomposition reaction is expressed as follows. AB molecule AB, breaks down to yield A atom, ion, or molecule A, plus B, atom, ion, or molecule B. It was the opposite of the original synthesis reaction. The decomposition reaction that occurs in your body are collectively referred to as catabolism. The decomposition reactions that occur in your body are collectively referred to as catabolism. Overall, catabolic reactions are usually exergonic because they release more energy than they absorb. For instance, the series of reactions that break down glucose to pyruvic acid with the net production of two molecules of ATP are important catabolic reactions in the body. These reactions will be discussed in Chapter 25. Exchange reactions. Many reactions in the body are exchange reactions. They consist of both synthesis and decomposition reactions. One type of exchange reaction works like this. AB plus CD yields AD plus BC. The bonds between A and B and B and C, the bonds between A and B and between C and D break, which is decomposition, and new bonds form. Synthesis between A and D and between B and C. An example of an exchange reaction is hydrochloric acid plus NaHCO3, sodium bicarbonate, yields H2CO3, carbonic acid plus NaCl, sodium chloride. Notice that the ions in both compounds have switched partners. The hydrogen ion, H, from HCl has combined with the bicarbonate ion, HCO3, from NaHCO3, and the sodium ion, Na plus, from NaHCO3 has combined with the chloride ion from hydrochloric, HCl. Reversible reactions. Some chemical reactions proceed in only one direction. Reversible reactions. Some chemical reactions proceed in only one direction, from reactants to products, as previously indicated by the single arrows. Other chemical reactions may be reversible. In a reversible reaction, the products can revert to the original reactants. A reversible reaction is indicated by two half arrows pointing in opposite directions. AB breaks down into A plus B, and A plus B combines to form AB. Some reactions are reversible only under special conditions, such as with water or heat. In that case, whatever is written above or below the arrows indicates the condition needed for the reaction to occur. In these reactions, AB breaks down into A and B only when water is added. An A and B reactor produce AB only when heat is applied. Many reversible reactions in the body require catalysts called enzymes. Often different enzymes guide the reactions into opposite directions. Inorganic compounds in solutions. The objective is to describe the properties of water and those of inorganic acid, bases, and salts. Also to distinguish among solutions, colloids, and suspensions, and to define pH and explain the role of buffer systems in homeostasis. Most of the chemicals in your body exist in the form of compounds. Biologists and chemists divide these compounds into two principal classes: inorganic compounds and organic compounds. Inorganic compounds usually lack carbon and are structurally simple. They include water and many salts, acids, and bases. Inorganic compounds may have either ionic or covalent bonds. Water makes up to 55 to 60% of lean adults' total body mass. Once again, water makes up 55 to 60% of a lean adult's total body mass. All other inorganic compounds combined add 1 to 2%. All other inorganic compounds combined add 1 to 2%. And examples of inorganic compounds that contain carbon are carbon dioxide, bicarbonate ion, and carbonic acid. Organic compounds always contain carbon, usually contain hydrogen, and always have covalent bonds. Most are large molecules, and many are made up of long chains of carbon atoms. Organic compounds make up the remaining 38 to 43% of the human body. Water. Water is the most important and abundant inorganic compound in all living systems. Although you might not be able to survive for weeks without food, without water you would die in a matter of days. Nearly all the body's chemical reactions occur in a watery medium. Water has many properties that make it such an indispensable compound for life. We've already mentioned the most important properties of water: its polarity, the uneven sharing of valence electrons that confer a partial negative charge near the one oxygen atom and two partial positive charges near the two hydrogen atoms in a water molecule. This property alone makes water an excellent solvent for other ionic or polar substances. It gives water molecules cohesion, the tendency to stick together, and allows water to resist temperature changes. Water as a solvent. In medieval times, people water as a solvent. In medieval times, people searched in vain for a universal solvent, a substance that would dissolve all other materials. They found nothing that worked as well as water. Although it is the most versatile solvent known, water is not the universal solvent sought by medieval alchemists. If it were, no container could hold it, because it would dissolve all potential containers. What exactly is a solvent? In a solution, substance called the solvent dissolves another substance called the solute. Usually there is more solvent than solute in a solution. For example, your sweat is a dilute solution of water, the solvent, plus small amounts of salts, the solutes. The versatility of water as a solvent for ionized or polar substances is due to its polar covalent bond and its bent shape, which allows each water molecule to interact with several neighboring ions or molecules. Solutes that are charged or contain polar covalent bonds are hydrophilic. Hydro meaning water, philic meaning loving, which means they dissolve easily in water. Common examples of hydrophilic solutes are sugar and salt. Molecules that contain mainly nonpolar covalent bonds, by contrast, are hydrophobic. Common examples of hydrophilic solutes are sugar and salt. Molecules that contain mainly Nonpolar covalent bonds, by contrast, are hydrophobic. They are not very water-soluble. Examples of hydrophobic compounds include animal fats and vegetable oils. To understand the dissolving power of water, consider what happens when a crystal of a salt, such as sodium chloride, is placed in water. The electronegative oxygen atom in water molecules attracts the sodium ions, and the electropositive hydrogen atoms in water molecules attracts the chloride ions. Soon, water molecules surround and separate. Sodium and chloride ions form. Breaking the ionic bonds that held NaCl together. In addition to dissolving sodium chloride, water also causes it to dissociate or separate into charged particles, which is discussed shortly. The electronegative oxygen atoms in water molecules attracts the sodium ions, and the electropositive hydrogen atoms in water molecules attract the chloride ions. Soon, water molecules surround and separate Na and Cl ions from each other at the surface of the crystal, breaking the ionic bonds that held NaCl together. The water molecules surrounding the ions also lessen the chance that Na and Cl will come together and reform an ionic bond. The ability of water to form solutions is essential to health and survival. Because water can dissolve so many different substances, it is an ideal medium for metabolic reactions. Water enables dissolved reactants to collide and form products. Water also dissolves waste products, which allow them to be flushed out of the body in the urine. Water and chemical reactions. Water serves as the medium for most chemical reactions in the body and participates as a reactant or product in certain reactions. During digestion, for example, decomposition reactions break down large nutrient molecules into smaller molecules by the addition of water molecules. This type of reaction is called hydrolysis. To loosen or break apart. Hydrolysis reactions enable dietary nutrients to be absorbed into the body. By contrast, when two smaller molecules join to form a larger molecule in a dehydration synthesis reaction, a water molecule is one of the products formed. As we will see later in the chapter, such reactions occur during synthesis of proteins and other large molecules. Thermal properties of water. In comparison to most substances, water can absorb or release a relatively large amount of heat with only a modest change in its own temperature. For this reason, water is said to have a high heat capacity. The reason for this property is the large number of hydrogen bonds in water. As water absorbs heat energy, some of the energy is used to break hydrogen bonds. Less energy is then left over to increase the motion of water molecules, which would increase the water's temperature. The high heat capacity of water is the reason it is used in automobile radiators. It cools the engine by absorbing heat without its own temperature rising an unacceptably high level. The large amount of water in the body has a similar effect. It lessens the impact of environmental temperature changes, helping to maintain the homeostasis of body temperature. Water also requires a large amount of heat to change from a liquid to a gas. Its heat of vaporization is high. As water evaporates from the surface of the skin, it removes a large quantity of heat, providing an important cooling mechanism. Water as a lubricant. Water is a major component of mucus and other lubricating fluids throughout the body. Lubrication is especially necessary in the chest, pleural and pericardial cavities, and abdomen, peritoneal cavity, where internal organs touch and slide over one another. It is also needed at joints where bones, ligaments, and tendons rub against one another inside the gastrointestinal tract, mucus, and other watery secretions moisten food, which aids their smooth passage through the digestive system. Solutions, colloids, and suspensions. A mixture is a combination of elements or compounds that are physically blended together but not bound by chemical bonds. For example, the air you are breathing is a mixture of gases that includes nitrogen, oxygen, argon, and carbon dioxide. These common liquid mixtures are solutions, colloids, and suspensions. Once mixed together, solutes in a solution remain evenly dispersed among the solvent molecules. Because the solute particles in a solution are very small, a solution looks clear and transparent. Por ejemplo, it's all about the segue, you know? You gotta get this stuff down. Okay. Hold on now. Where was I? Okay. A colloid. A colloid differs from a solution mainly because of the size of its particles. The solute particles in a colloid are large enough to scatter light, just as water droplets in fog scatter light from a car's headlight beams for this reason. Colloids usually appear translucent or opaque. Milk is an example of a liquid that is both a colloid and a solution. The large milk proteins make it a colloid, whereas the calcium, salts, milk, sugar, lactose, ions, and other small particles are in solution. The solutes in both solutions and colloids do not settle out and accumulate on the bottom of the container. In a suspension, by contrast, the suspended material may mix with the liquid or suspending medium for some time, but eventually it will settle out. Blood is an example of a suspension. When freshly drawn from the body, blood has an even reddish color. After blood sits for a while in a test tube, red cells settle out of the suspension and drift to the bottom of the tube. The upper layer, the liquid portion of blood, appears pale yellow and is called blood plasma. Blood plasma is both a solution of ions and other small solutes in a colloid due to the presence of large plasma proteins. The concentration of a solution may be expressed in several ways. One common way is by a mass per volume percentage, which gives the relative mass of a solute found in a given volume of solution. For example, you may have seen the following on the label of a bottle of wine: alcohol 14.1% by volume. Another way expresses concentration in units of moles per liter, or mol/L, moles per liter, which relate to the total number of molecules in a given volume of solution. A mole is the amount of any substance that has a mass in grams equal to the sum of the atomic masses of all of its atoms. For example, 1 mole of the element chlorine is 35.45 grams, and 1 mole of the salt sodium chloride is 58.44 grams. Just as a dozen always means 12 of something, a mole of anything has the same number of particles. 6.023 times 10 to the 23rd power. This huge number is called Avogadro's number. So let's read that one again. A mole is the amount of any substance that has a mass in grams, equal to the sum of the atomic masses of all of its atoms. For example, 1 mole of the element chlorine with an atomic mass equals 35.45 is 35.45 grams, and 1 mole of the salt sodium chloride, NaCl, is 58.44 grams. 22.99 for the sodium, and 35.45 for the chloride. Just as a dozen always means 12 of something, a mole of anything has the same number of particles, 6.023 times 10 to the 23rd power. This huge number is called Avogadro's number. Thus, measurements of substances that are stated in moles tell us about the number of atoms, ions, or molecules present. This is important when chemical reactions are occurring because each reaction requires a set number of atoms of specific elements. And there's a table. Definition: percentage, mass per volume, the number of grams of a substance per 100 milliliters or milliliters of solution. Example: to make a 10% NaCl solution, take 10 grams of NaCl and add enough water to make a total of 100 milliliters of solution. Molarity equals moles, mole per liter. A 1 molar or 1 m solution equals 1 mole of a solution and 1 liter of a solution. To make a 1 mole 1 m solution of NaCl, dissolve 1 mole of NaCl, 58.44 grams, in enough water to make a total of 1 liter of solution. Inorganic acids, bases, and salts. When inorganic acids, bases, or salts dissolve in water, they dissociate. That is, they separate into ions and become surrounded by water molecules. An acid is a substance that dissociates into one or more hydrogen ions and one or more anions. Because hydrogen is a single proton with one positive charge, an acid is also referred to as a proton donor. A base, by contrast, removes hydrogen from a solution and is therefore a proton acceptor. Many bases dissociate into one or more hydroxide ions and one or more cation. A salt, when dissolved in water, dissociates into cations and anions, neither of which is hydrogen or OH. In the body, salts such as potassium chloride are electrolytes that are important for carrying electrical currents. Ions flowing from one place to another, especially in nerve and muscular tissue. The ions of salts also provide many essential chemical elements in intracellular and extracellular fluids such as blood, lymph, and interstitial fluid of tissue. Acids and bases react with one another to form salts. For example, the reaction of hydrochloric acid, HCl, and potassium hydroxide, KOH, a base, produces the same potassium chloride, KCl, and water, H2O. This exchange reaction can be written as follows. HCl, which is the acid, plus KOH, which is the base, yields hydrogen, plus chloride, plus potassium, plus hydroxide, to yield KCl plus H2O, or salt plus water. Acid-base balance, the concept of pH. To ensure homeostasis, intracellular and extracellular fluids must contain almost balanced quantities of acids and bases. The more hydrogen ions dissolved in a solution, the more acidic the solution, the more hydroxide ions, the more basic, alkaline. The solution. I read that incorrectly. The more hydrogen ions dissolved in a solution, the more acidic the solution. The more hydroxide ions, the more basic the solution. The chemical reaction that takes place in the body are very sensitive to even small changes in acidity or alkalinity of the body fluids in which they occur. Any departure from the narrow limits of normal hydrogen and hydroxide concentrations greatly disrupts body functions. A solution's acidity or alkalinity is expressed on the pH scale, which extends from 0 to 14. This scale is based on the concentration of hydrogen in moles per liter. A pH of 7 means that a solution contains 1/10 millionth of a mole of hydrogen ions per liter. Let's read that again. A solution's acidity or alkalinity is expressed on a pH scale which extends from 0 to 14. This scale is based on the concentration of hydrogen in moles per liter. A pH of 7 means that a solution contains 1/10,000,000th of a mole of hydrogen ions per liter. The number 0.0000001 is written as 1 times 10 to the negative 7 in scientific notation, which indicates that the number is 1 with the decimal point moved 7 places to the left. To convert this value of pH, the negative exponent, negative 7, is changed to a positive number, 7. A solution with a hydrogen concentration of 0.0001 or 10 to the negative 4 moles per liter has a pH of 4. A solution with a hydrogen concentration of 0.0000000001 moles per liter has a pH of 9, and so on. It is important to realize that a change of one whole number on the pH scale represents a tenfold change in the number of hydrogen. A pH of 6 denotes 10 times more hydrogen than a pH of 7, and a pH of 8 indicates 10 times fewer hydrogen than a pH of 7, and 100 times fewer hydrogen than a pH of 6. The midpoint of the pH scale is 7. When the concentration of hydrogen and hydroxide are equal, a substance with a pH of 7, such as pure water, is neutral. A solution that has more hydrogen than hydroxide is an acidic solution and has a pH below 7. A solution that has more hydroxide than hydrogen is basic, alkaline solution and has a pH above 7. Maintaining pH buffer systems is the next chapter. The chapter is maintaining pH buffer systems. Although the pH of body fluids may differ, as we have discussed, the normal limits for each fluid are quite narrow. Homeostatic mechanisms maintain the pH of blood between 7.35 and 7.45, which is slightly more basic than pure water. You will learn in Chapter 27 that if the pH of blood falls below 7.35, a condition called acidosis occurs, and if the pH rises above 7.45, it results in a condition called alkalosis. Both conditions can seriously compromise homeostasis. Saliva is slightly acidic, and semen is slightly basic. Because the kidneys help remove excess acid from the body, urine can be quite acidic. Even though strong acids and bases are continually taken into and formed by the body, the pH of fluid inside and outside cells remains almost constant. One important reason is the presence of buffer systems, which functions to convert strong acids or bases into weak acids or bases. Strong acids or bases ionize easily and contribute many hydrogen or hydroxide to a solution. Therefore, they can change pH drastically, which can disrupt the body's metabolism. Weak acids or bases do not ionize as much and contribute fewer hydrogen or hydroxide. Hence, they have less effect on the pH. The chemical compounds that can convert strong acids or bases into weak ones are called buffers. They do so by removing or adding protons. One important buffer system in the body is the carbonic acid bicarbonate buffer system. Carbonic acid, H2CO3, can act as a weak acid, and the bicarbonate ion, HCO3, can act as a weak base. Hence, this buffer system can compensate for either an excess or a shortage of hydrogen. For example, if there is an excess of hydrogen in an acidic condition, HCO3 can function as a weak base and remove the excess hydrogen as follows. The hydrogen ion plus bicarbonate ion, the weak base, will yield carbonic acid, H2CO3. If there is a shortage of hydrogen, an alkaline condition, by contrast, H2CO3 can function as a weak acid and provide needed hydrogen as follows. Carbonic acid, a weak acid, will yield hydrogen ion plus bicarbonate ion. In Chapter 27, we'll describe buffers and their role in maintaining the acid-base balance in more detail. Just so that you get the idea, the pH values of selected substances are discussed. Gastric juice, found in the stomach, has a pH of 1.2 to 3. Lemon juice is 2.3. Vinegar, 3.0. Carbonated soft drink, 3.0 to 3.5. Orange juice, 3.5. Vaginal fluid, 3.5 to 4.5. Tomato juice is 4.2. Coffee is 5.0. Urine is 4.6 to 8.0. Saliva 6.35 to 6.85. Milk is 6.8. Distilled pure water is 7.0. Blood is 7.35 to 7.45. Semen is 7.2 to 7.6. Cerebral spinal fluid associated with the nervous system is 7.4. Pancreatic juice, digestive juice of the pancreas, 7.1 to 8.2. Bile, liver secretion that aids fat digestion, is 7.6 to 8.6. Milk of magnesia is 10.5, and lye, sodium hydroxide, is 14. Next, we will discuss organic compounds. The objective of this chapter is to describe the functional group of organic molecules, identify the building blocks and functions of carbohydrates, lipids, and proteins, describe the structure and function of deoxyribonucleic acid, DNA, ribonucleic acid, and adenosine triphosphate, ATP. Inorganic compounds are relatively simple. Their molecules have only a few atoms and cannot be used by cells to perform complicated biological functions. Many organic molecules, by contrast, are relatively large and have unique characteristics that allow them to carry out complex functions. Important categories of organic compounds include carbohydrates, lipids, proteins, nucleic acids, and adenosine triphosphate, also known as ATP. Carbon and its functional groups. Carbon has several properties that make it particularly useful to living organisms. For one thing, it can form bonds with 1 to thousands of other carbon atoms to produce large molecules that can have many different shapes. I want to read that again. Carbon has several properties that make it particularly useful to living organisms. For one thing, it can form bonds with 1 to thousands of other carbon atoms to produce large molecules that can have many different shapes. Due to this property of carbon, the body can build many different organic compounds, each of which have a unique structure and function. Moreover, the large size of most carbon-containing molecules and the fact that some do not dissolve easily in water make them useful materials for building body structures. Organic compounds are usually held together by covalent bonds. Carbon has 4 electrons in its outermost valence shell. It can bond covalently with a variety of atoms, including other carbon atoms, to form rings and straight or branched chains. Other elements that most often bond with carbon in organic compounds are hydrogen, oxygen, and nitrogen. Sulfur and phosphorus are also present in organic compounds. The other elements listed are present in smaller numbers of organic compounds. The name and structural formula, and the occurrence and significance A hydroxyl group, ROH, alcohols contain an OH group, which is polar and hydrophilic due to its electronegativity, the electronegative oxygen atom. Molecules with many hydroxyl groups dissolve easily in water. Sulfhydryl thiols have an SH group which is polar and hydrophilic due to its electronegative sulfur atom containing amino acids. The building blocks of protein contain SH groups which help stabilize the shape of proteins. An example is the amino acid cysteine. Carbonyl. Ketones contain a carbonyl group within the carbon skeleton. The carbonyl group is polar and hydrophilic due to its electronegative oxygen atom. Aldehydes have a carbonyl group at the end of the carbon skeleton. Carboxyl. Carboxylic acid contains a carboxyl group at the end of the carbon skeleton. All amino acids have a carboxyl group at one end. The negatively charged form predominates at the pH of body cells and is hydrophilic. Ester. Esters predominate in dietary fats and oils and also occur in our body's triglycerides. Aspirin is an ester of salicylic acid, a pain-relieving molecule found in the bark of the willow tree. Phosphate. Phosphates containing a phosphate group, which is very hydrophilic due to the dual negative charges. An important example is adenosine triphosphate, ATP, which transfers chemical energy between organic molecules during chemical reactions. Amines have an NH2 group which can act as a base and pick up a hydrogen ion, giving the amino group a positive charge. At the pH of body fluids, most amino groups have a charge of 1 positive. All amino acids have an amino group at one end. To recap, the major functional groups, name and structural formula are hydroxyl, sulfhydryl, carbonyl, carboxyl, ester, phosphate, and amino. The chain of carbon atoms in an organic molecule is called the carbon skeleton. The chain of carbon atoms in an organic molecule is called the carbon skeleton. Many of the carbons are bonded to hydrogen atoms, yielding a hydrocarbon. Also attached to the carbon skeleton are distinctive functional groups, other atoms or molecules bound to the hydrocarbon skeleton. Each type of functional group has a specific arrangement of atoms that confers characteristic chemical properties on the organic molecule attached to it. Because organic molecules often are big, there are shorthand methods for representing their structural formula. Small organic molecules can combine into very large molecules that are called macromolecules. Macromolecules are usually polymers. A polymer is a large molecule formed by the covalent bonding of many identical or similar small building blocks called monomers. Usually, the reaction that joins two monomers is a dehydration synthesis. In this type of reaction, a hydrogen atom is removed from one monomer and a hydroxyl group is removed from the other to form a molecule of water. Macromolecules such as carbohydrates, lipids, proteins, and nucleic acids are assembled in cells via dehydration synthesis reactions. Molecules that have the same molecular formula but different structures are called isomers. For example, The molecular formula for sugar, glucose, and fructose are both C6H12O6. The individual atoms, however, are positioned differently along the carbon skeleton, giving the sugar different chemical properties. Carbohydrates include sugars, glycogen, starches, and cellulose. Even though there are large and diverse groups of organic compounds and have several functions, carbohydrates represent only 2 to 3% of your total body mass. In humans and animals, carbohydrates function mainly as a source of chemical energy for generating adenosine triphosphate needed to drive metabolic reactions. Only a few generating ATP needed to drive only a few carbohydrates are used for building structural units. One example is deoxyribose, a type of sugar that is a building block of deoxyribonucleic acid, the molecule that carries inherited genetic information. I'm going to reread that just so that it is more understandable. Carbohydrates include sugar, glycogen, starches, and cellulose. Even though they are a large and diverse group of organic compounds and have several functions, carbohydrates represent only 2 to 3% of your total body mass. In humans and animals, carbohydrates function mainly as a source of chemical energy for generating adenosine triphosphate needed to drive metabolic reactions. Only a few carbohydrates are used for building structural units. One example is deoxyribose, a type of sugar that is a building block of deoxyribonucleic acid, DNA, the molecule that carries inherited genetic information. Carbon, hydrogen, and oxygen are the elements found in carbohydrates. The ratio of hydrogen to oxygen atoms is usually 2 to 1, the same as in water. Although there are exceptions, carbohydrates generally contain one water molecule for each carbon atom. Although there are exceptions, carbohydrates generally contain one water molecule for each carbon atom. This is the reason they are called carbohydrates, which means watered carbon. The three major groups of carbohydrates based on their size are monosaccharides, disaccharides, and polysaccharides. Monosaccharides are simple sugars that contain from 3 to 7 carbon atoms. An example would be glucose, the main blood sugar, fructose found in fruits, galactose in milk sugar, deoxyribose in DNA, and ribose in RNA. Disaccharides are simple sugars formed from the combination of two monosaccharides by the dehydration synthesis. Sucrose, table sugar, glucose plus fructose. Lactose, which is milk sugar, equals glucose plus galactose. And maltose equals glucose plus glucose. Polysaccharides from tens to hundreds of monosaccharides joined by dehydration synthesis. Glycogen, the stored form of carbohydrates in animals. Starch, the stored form of carbohydrates in plants and main carbohydrate in food. Cellulose, part of cell walls in plants that cannot be digested by humans but aids movement of food through intestines. Monosaccharides and disaccharides, the simple sugars. Monosaccharides and disaccharides are known as simple sugars, the monomers of carbohydrates. Monosaccharides contain from 3 to 7 carbon atoms. They are designated by names ending in "-ose" with a prefix that indicates the number of carbon atoms. For example, monosaccharides with 3 carbon are called trioses. There are also tetroses, 4 carbon sugars, pentoses, 5 carbon sugars, hexoses, 6 carbon sugars, and heptoses. Examples of pentoses and hexoses are illustrated in 2.14. Cells throughout the body break down the hexose glucose to produce ATP. A disaccharide is a molecule formed from the combination of two monosaccharides by the dehydration synthesis. For example, molecules of the monosaccharide glucose and fructose combine to form a molecule of disaccharide sucrose, table sugar. Glucose and fructose are isomers. As you learned earlier in the chapter, isomers have the same molecular formula, but the relative positions of oxygen and carbon atoms are different, causing the compounds to have different chemical properties. Notice that the formula for sucrose is C12H22O11, not C12H24O12, because a molecule of water is removed as the two monosaccharides are joined. Disaccharides can also be split into smaller, simpler molecules by hydrolysis. The molecule of sucrose, for example, may be hydrolyzed into its components, glucose and fructose, by the addition of water. Some individuals use artificial sweeteners to limit their sugar consumption for medical reasons, while others do it to avoid calories that might result in weight gain. Artificial sweeteners are much sweeter than sucrose, have fewer calories, and do not cause tooth decay. Polysaccharides. The third major group of carbohydrates is the polysaccharides. Each polysaccharide molecule contains tens or hundreds of monosaccharides joined through dehydration synthesis reactions. Unlike simple sugars, polysaccharides usually are insoluble in water and do not taste sweet. The main polysaccharides in the human body is glycogen, which is made entirely of glucose monomers linked to one another in branching chains. The main polysaccharide in the human body is glycogen, which is made entirely of glucose monomers linked to one another in branching chains. A limited amount of carbohydrates is stored as glycogen in the liver and skeletal muscles. Starches are polysaccharides formed from glucose by plants. They are found in foods such as pasta and potatoes and are major carbohydrates in the diet. Like disaccharides, polysaccharides such as glycogen and starches can be broken down into monosaccharides through hydrolysis reactions. For example, when the blood glucose level falls, liver cells break down glycogen into glucose and release it into the blood, making it available to body cells which break it down to synthesize ATP. Cellulose is a polysaccharide formed from glucose by plants that cannot be digested by humans but does provide bulk to help eliminate feces. Lipids. A second important group of organic compounds is lipids. Lipid fats make up 18 to 25% of body mass in lean adults. Like carbohydrates, lipids contain carbon, hydrogen, and oxygen. Unlike carbohydrates, they do not have a 2-to-1 ratio of hydrogen to oxygen. The proportion of electronegative oxygen atoms in lipids is usually smaller than in carbohydrates, so there are fewer polar covalent bonds. As a result, most lipids are insoluble in polar solvents such as water. They are hydrophobic. Because they are hydrophobic, only the smallest lipids, some fatty acids, can dissolve in watery blood plasma. To become more soluble in blood plasma, other lipid molecules join with hydrophilic protein molecules. The resulting lipid-protein complexes are termed lipoproteins. Lipoproteins are soluble because the proteins are on the outside of the lipids, and the lipids are on the inside. Once again, lipoproteins are soluble because the proteins are on the outside and the lipids are on the inside. The diverse lipid family includes fatty acids, triglycerides, fats and oils, phospholipids, lipids that contain phosphorus, steroids, lipids that contain rings of carbon atoms, eicosanoids, 20 carbon lipids, and a variety of other lipids including fat-soluble vitamins, vitamins A, D, E, and K, and lipoproteins. Fatty acids. Among the simplest lipids are the fatty acids, which are used to synthesize triglycerides and phospholipids. Fatty acids can also be catabolized to generate adenosine triphosphate. A fatty acid consists of a carboxyl group Among the simplest lipids are the fatty acids, which are used to synthesize triglycerides and phospholipids. Fatty acids can also be catabolized to generate adenosine